First, we are going to talk about the base pairing of DNA nucleotides. Purines have two rings and include adenine and guanine. Pyrimidines have one ring and include thymine and cytosine. Pyrimidines bond to purines. A and T bond together with 2 hydrogen bonds, and C and G bond together with 3. I have decided to build the DNA model using candy. There is a key in the top that tells you which lifesaver color corresponds to which nucleotide. The main takeaway is that the hydrogen bonds that connect the base pairs are important to stabilizing DNA because they hold the complementary strands of DNA together. Base stacking is another important concept in the DNA model. Base stacking refers to the way that the purine and pyrimidine bonds are stacked inside of the DNA backbone as seen in the picture. They have van der Waals interactions that stabilize them, which are dependent on distance. Because they are stacked close enough, they share electron clouds, forming a bond. Next, we will talk about the ribose carbon of DNA, which is a deoxyribose. It has a hydrogen at the 2' position instead of hydroxyl. The carbons are labeled in a clockwise direction from 1 to 5. The nitrogenous bases are attached to the 1' carbon of the deoxyribose, and it is a glycosidic bond. The deoxyribose is represented by the pentagon of marshmallows. The toothpick sticking out is a 5' carbon. The strands are antiparallel, so the sugars are oriented in opposite directions. Phosphates bind to the sugars to create a sugar-phosphate backbone. These bonds help create the framework of DNA and play a huge role in the shape. The phosphates are represented by the Skittles. The phosphate is connected to the 5' carbon of one sugar and the 3' carbon of the next. The phosphate-sugar bond is called a phosphodiester bond, and it helps create the sugar-phosphate backbone. DNA has major and minor grooves. The major grooves are where the backbones are far apart, and the minor grooves are where the backbones are close together. If a DNA strand is right-handed, then the part of the backbone that is in front points up and to the right. If the DNA is left-handed, the backbone that is in the front goes up and towards the left. The phosphate backbone of DNA contains a negative charge, which destabilizes the DNA molecule. Negative charges work to push the strands away from one another despite all the forces we talked about. When DNA is placed in a salt solution, the negative charges of the backbone can be neutralized by the salt ion. Tm refers to the melting temperature. In a low salt concentration, DNA will have a lower Tm than in a high salt concentration. Tm increases as salt concentration increases. Melting can also be referred to as denaturation. If there is a low salt concentration, there is not much shielding of the negative charges, which are working to pull apart the DNA. Here are my sources.